I wanted to start today just to back up with meiosis. So again, the purpose of meiosis is to make sperm and egg, but we also have to start with our sperm and egg. So this is a sperm that I've indicated in blue to represent the male. You can see that it has two chromatids in it. The only organelle that a sperm carries is a centriole because it needs to be lightweight and to travel. Whereas an egg will have, this one also has two chromatids in it, and this egg has all of the organelles from the mother. Because organelles are costly and take a lot of energy to create, so all the organelles are passed on through your mother's line. Scientists specifically look at mitochondrial DNA that's found in the mitochondria, and we can trace back lineages because your mitochondria you receive from your mother. So at fertilization, we would have our two chromatids from the mother and our two chromatids here from our father. They're going to duplicate during the S phase. So these are identical matches of each side. So these are sister chromatids basically. These two are now called homologous chromosomes. Homologous meaning they are same. So this is the same chromosome for hair color, eye color, for example, from the mother. And again, those same genes are on the one for the father. And they are in the same order as well. So again, starting from our egg, you'll notice that these chromosomes do not have any other colors mixed in with them. You can see this half is from mom and this half is from dad. So we are leaving interphase. We're going to enter into meiosis I. It's very important when you're talking about meiosis is that you include Roman numerals, because if you just said prophase, we would think that we would be in mitosis. Again, the purpose of meiosis is to make gametes, sperm and eggs, that have half of the genetic information. So we are in prophase 1 of meiosis. The chromosomes have lined up with their homologous chromosome, meaning it's lined up with the one from the mother or the father. And we have something called crossing over that takes place. So these are mirror images of each other. So you can see here that this little tip has broken off and has gone over to here. And again, these are mirror images. So likewise, these two tips have broken off and they've moved over here. This allows for genetic variation. As we leave prophase 1, we are going to enter into metaphase. Metaphase is very similar in mitosis. Again, this is metaphase 1. Metaphase means middle. We have the law of independent assortment because this chromosome could be on this side and this chromosome could have been on that side. So you can see all 2 to the 23rd power gives us over 8 million possibilities for genetic variation. So as we enter into anaphase 1, you can see that we are splitting the homologous chromosomes apart. With our spindle fiber and our centrioles here, we are pulling them apart. We're going to enter into prophase 2, and we end up with two separate cells here. We're going to enter into prophase 2, which means we're going to have our nuclear membrane rebounds. There is no interphase 2, so keep that in mind. As we speed ahead over here, we're going to look at meiosis II. We have our chromosomes. They are now lining up again in metaphase Roman numeral 2. They are lining up along the middle. Again, our centrals come in with our spindle fibers, and we are going to start to divide those. So we've pulled them apart in anaphase 2, and we've also gone through telophase 2 as well as cytokinesis. So we've ended up with 4 cells. You can see that this has the half of the genetic information from when we started. Now, in males, each one of these will turn into a sperm. So you can see a lot of genetic variation going on over here. Okay. Whereas in females, those organelles, again, are very expensive to make and a lot of energy. So one of those becomes the egg or ovum. You can see here's the DNA of the mitochondria is here. All those other organelles are here. These three are called polar bodies, and usually they're just absorbed by the egg for nutrients. So now this is an egg. It has half the genetic information, and it's going to hang out in the ovary until ovulation takes place. It will then later on join up with a sperm. Feel free to pause the video to get the big picture of the graphic organizer.